Okay, I was thinking that for all the biomanians, all the people who are watching this show right now, that we kind of need a theme song. And maybe I'll just make one up right now, like, it's time to learn biology. Yeah, it's time to learn biology. 2, 3. AP Bio review for the next hour. That's what we're gonna do. Yeah, it's time to review biology. Okay, Sunday night, and here we are broadcasting from the world headquarters of learn-biology.com, located in Berkeley, California. It is Mr. W bringing you another live review about AP Biology. We are heading at breakneck speed towards the AP Bio exam, but there is still so much time. You can completely change your experience of the test. All you've got to do is review. And I actually want to suggest that the best way to do that is not just with me. I mean, I am so happy that you are here with me, but to get all the additional resources that you wind up getting if you subscribe to learn-biology.com. One of them is something that you could experience as quickly as tomorrow night, which is I'm also doing reviews on Zoom, which are just much nicer and they're much warmer. I love this. But the channel through which you can give me feedback is fairly narrow. So I love Zoom because it's really a kind of virtual presence that I want to just encourage you to join into. Lily, I am so glad to see you here. And I know that there are other people here as well. And I'm looking forward to hearing people say hi and tell me where they are and who they are. And it's just great to have you with me tonight. I know that there's a lot of other ways to review, and the fact that you're here with me really, really means a lot. So, let's go on. Let's talk about tonight's agenda. And I'm realizing I didn't completely update this slide, but we'll do our best. So we have done everything on this list. And in fact, we've taken care of eukaryotic gene expression too. I'm going to turn on the pen. I'm going to cross that out. We also did mutation last night. So what's left? Just horizontal gene transfer and biotechnology. So that's what's on the docket for tonight. That's what we're going to be looking at. So I'll do a presentation, but I also want to emphasize my main agenda at these review sessions is to answer your questions. And to the extent that I'm able, if you have a question, you can put it in the comment thread. I'll probably get to the end of what I'm talking about, and then I will address it. And I'll address it in one of two ways. One is I'll just tell you what I think I can do to help you out in terms of your question. And the other is, if that's not possible, then what I'll do is I'll handle it at the next session or something like that. So anyhow, once again, I want to thank you very much for being here. And let's make this look a little bit better by me going to a better view, which involves me hitting one of these buttons. I haven't quite mastered which one. I think it's, no, that one makes me super small. I don't like that one. No, I want to be off the screen. This one, fantastic. So here I am, and this is a good opportunity for me to introduce myself in my little virtual classroom. I'm Glenn Wokenfeld, also known as Mr. W. You've probably heard my story before, but I'm a relatively recently retired AP biology teacher, retired from Berkeley High School, where I spent many, many beloved years. And now I'm teaching you through this medium, through my Science Music Videos YouTube channel and through my learn-biology.com website. When I taught AP Bio, I had a lot of success in getting students to get 4s and 5s on the AP Bio exam. And I believe that if you stick with me, through these reviews, but more importantly, through my website, learn-biology.com, which does require a $24.95 monthly subscription. And you know, that could add up to potentially a lot over the course of a year, but you're only going to do it for the next 10 days, and then you're going to cancel. So really, your total cost is $24.95, and it's the best value available in biology education. We'll actually use it tonight, and you'll see how it can totally transform your experience. And I know that there are people who are watching now who haven't subscribed yet because, you know, I just know it, and I really want to encourage you to do it because it's going to be that thing that will change the game for you in terms of understanding the greatest subject of all, biology. And if you can't spell that, you should learn how to spell it before now and the AP Bio exam, because that's biology. I had so much success with my students for a couple of reasons. You know, I sing songs and things like that, and that's fun. But I'm also the author of the learn-biology.com website. It's a full-fledged biology curriculum. It's endorsed by the college board insofar as you could, a teacher, if you were a teacher, you could use it in lieu of a textbook. And it's got everything you need for learning biology. It's got tutorials, it's got quizzes, it's got interactions, it's got games, and above all for you, since we're heading towards the AP Biology exam, it's got comprehensive AP reviews that, again, are going to help you understand so much biology. There is so much time. If you spent an hour a night on Learn-Biology, you'd learn so much. I also have an app that is deeply connected to the website. It has a lot of similar content, and you can do, what do we got here? Here's the main menu. Here's an FRQ. Here's a multiple choice question. And here's a game called the Tap Challenge. Super fun, super interactive. And I just can't encourage you enough to go to learn-biology.com. If you do that, just hit the student button. I'll give you the link for going more directly to our student page in a moment. One thing you should be doing is you should download and use our checklist. We have a checklist that helps you go through the entire course and self-assess. What you want to be doing right now as you prepare for the AP Bio exam is you want to be identifying the gaps in your knowledge and then filling them. You can use the checklist. You can use multiple choice quizzes to identify your gaps. I explained how to do that in a video that's like your plan for AP Bio. You'll find it on Science Music Videos if you go there. But you find the gaps and then you fill them. Find the gaps and you fill them. That's the recursive cycle that you go through. And you need sort of a plan to do that, and that's why I put this checklist together. So hopefully you've downloaded it. If you haven't downloaded it, download it now. If you're a subscriber to learn-biology.com, you probably haven't. So anyhow, let's start the content of tonight's show. Thank you for patiently waiting for me. I haven't seen anybody really chat in saying hello other than Lily. And Lily, I want you to know I really appreciate that. But what we've got on the docket tonight, again, horizontal gene transfer and biotechnology. Here are some of the questions we'll be addressing in this section. We'll be contrasting horizontal gene transfer with vertical gene transfer. Horizontal gene transfer is something that we humans and in general we animals don't do. We do vertical gene transfer. I'll show you a diagram about that in a minute. Describe bacterial transformation, bacterial conjugation, and viral transduction. And finally, we'll learn about viral recombination. I'm looking down because a piece of my microphone just fell off. It's a little bit disorienting. Just give me a moment. Okay. Well, I think that that is not going to be found. Maybe I'll go back and delete this moment of the show. But how's the sound? Sound okay, guys? You can hear me okay? Everybody out in internet land? I'm audible. I'm hearable. I sure hope so. I'm going to go on. I'm going to go on with this lesson, and let's go ahead and do this. So, all right. Horizontal gene transfer. How's it different from vertical gene transfer? Vertical gene transfer is shown here, and it's the kind of thing that we eukaryotes are used to doing. It's the transmission of all or half of their genome between parents to offspring. If we were asexual reproducers, it would be 100%. And it's not only bacteria and some single-celled organisms that do that. You know, there are species of lizards that are all female. They reproduce by parthenogenesis. That's where the eggs develop on their own without being fertilized. So if you were a female parthenogenetic lizard, you would just pass on all of your genome onto all of your offspring. That's a form of vertical gene transfer. In the kind of sexual reproduction that most eukaryotes do, including, of course, humans, we pass on half of our genomes. We undergo meiosis, and a male passes on half of his genome through the sperm, the female through the egg, and that's basically what happens. Well, that's vertical gene transfer. It's what we think of when we think about transmission of genes. Horizontal gene transfer is something that we don't do, but I think it would be a great source of a science fiction series, because imagine that you go up to someone and you touch them, and as soon as you touch them, some of your genes go to them, and they start expressing some of your genetic traits. Wow, that's kind of a weird idea, but that's really what it's all about. Vampires are kind of like that, because but there's no vampires. Anyhow, horizontal gene transfer, one organism transfers genes to another organism that is not its offspring. And in unicellular organisms, the newly acquired genes become part of the recipient's genome, and they get passed on to its offspring. And in a multicellular recipient, there's only long-lasting results if the genes are transferred into the germline. And I want to actually adjust something that I said before. In multicellular organisms like us, we don't do horizontal gene transfer, but we've kind of been, in a weird way, the victim of it. Through viruses, we can receive genes from other organisms. So I'll explain that a little bit further on down the line. I again want to invite people to interrupt me at any moment, and just, of course, you can't like interrupt me like shouting and say, "Hey, Mr. W." But what you can do is you can put a comment in the chat if anything that I say sounds confusing, if you need further explanation, if it sparks some curiosity. I know that we get fairly mercenary at this point in the year, where it's like, okay, is it on the test? Horizontal gene transfer is on the test. Diagrams like this have been on the test, and that's why I'm covering it right now. But, you know, if there's just something that you're curious about, let me know, and I'll address it. So here's one form of horizontal gene transfer. It's called bacterial conjugation. And the basic idea is that bacteria have, in addition to their main chromosome, small circles of DNA that are called plasmids. Now, of course, in terms of size, this isn't correctly represented, but a bacterial chromosome might have 3 million bases, and a plasmid might have 20,000 bases. So, you know, 20,000, 3 million, this would be much, much bigger in terms of the amount of DNA. The plasmids express genes for a membrane extension called a pilus. So that's this extension. And that pilus, when it contacts a second cell, enables the plasmid to be copied and transmitted into the recipient. At that moment, the recipient receives all the genes that are on the plasmid. So the recipient, cell number 2 here, has been genetically transformed through its receipt of genetic material from the original cell. Let's call it the donor cell. Now, that ain't something that humans do, and this is something that happens all the time in bacteria, and it plays a key role in the spread of antibiotic resistance. Yera, thank you so much for asking this question about why this is done. This is done for a couple of reasons, but you sort of have to do a little bit of reset when you're thinking about biology, and you're thinking about like the why of things. The plasmids are genes. The plasmids are genes who have their own purpose, and their purpose is to replicate themselves. So when a plasmid passes itself on to another cell, it's achieved evolutionary success. Fitness, evolutionary fitness, is about the ability to pass on your genetic information and have that genetic information survive in your descendants. A plasmid is doing that on a very, very small level, but like the plasmid is doing what plasmids do. It's replicating itself. It's passing itself on. So you can think of it from the perspective of the bacterial species, and in some ways, like it has a positive effect, but really think of it in terms of the plasmid. The plasmid is a piece of DNA. It wants to replicate itself. Conjugation is how it replicates itself. So I hope that's a satisfying answer. Let's go on. Another form of horizontal gene transfer is called transformation. We're going to meet transformation again in just a minute when we look at biotechnology. Actually, we'll meet plasmids again when we talk about biotechnology. So in bacterial transformation, here's what happens. This is showing a cell dying and disintegrating, and as it's disintegrating, it's releasing some of its DNA into the environment. Some of those DNA fragments can actually be in the form of fragments. Now, what happens is that cell B over here, which is a living cell, it can pick up some of the DNA from the environment. There's a variety of mechanisms that can make that happen. It doesn't happen at a very high rate, but if you're doing genetic engineering, you can kind of goose the rate, you can increase the rate so that transformation happens more easily. If you've done a lab called the PIGLO lab or a transformation lab, that's what you did. What you did was you abused a bacterial cell a little bit. You abused its membrane by adding transformation solution, a salt, or you've done this thing called heat shocking. And what that does is it makes the membrane more permeable to taking up genetic material. Well, then that genetic material can then exist as a plasmid, or it can integrate itself into the genome of the recipient. So another form of horizontal gene transfer. This cell took up or acquired new genes, not from its parents, but from the environment. So now we've talked about conjugation and we've talked about transformation, two forms of horizontal gene transfer. So any other questions now that we've talked about conjugation and bacterial transformation? I know there's a little bit of a time lag, but let's go on. The DNA can include plasmids, and as I said before, this can include transformation. It's a big deal in bacterial transformation. And Maxime, thank you for telling me, because knowing that there are not questions is super useful too. So thank you very much, Maxime. All right. There's another form of horizontal gene transfer, and that's called transduction. Transduction involves horizontal gene transfer through viruses. Here's what happens. During viral infections, what happens, here's a virus landing on a bacterial cell. During the replicative cycle of a virus, viruses inject their genetic material. They wind up breaking down the genetic material of their host. They use the host's genetic machinery to create new viruses. A virus is kind of like an intracellular pirate. Well, when that happens, new viruses get assembled as from pieces of new viral genetic material and new viral proteins that have been created inside the doomed cell that was attacked. Well, sometimes the DNA fragments that get picked up were from the host cell. They're not viral DNA, but they're host pieces of DNA. When the virus attacks another cell in another organism, it can bring in that other organism's DNA. So I've color-coded this to make it remotely clear. This was the genetic material of the host. Here it is broken down. The virus picks up some of that genetic material, and now it's carrying it to another cell. And now that cell is being infected with that new genetic material, and it winds up incorporating it into its genome. So this is a very indirect route of horizontal gene transfer, but over evolutionary time, it can be quite significant. And we have in our genome many pieces of DNA that are kind of foreign to our species, and they came to us through infection by viruses that infected our germ cells. Kind of unbelievable, but a significant part of our genome. I don't know the percentage, but it's not 1%. It's something that's much higher than this. And Yera, what a Class A gold medal question. In order for this to happen in eukaryotes like us, in order to have the DNA from other species enter our genome through a viral infection, it's got to be a retrovirus. Retroviruses are viruses that have RNA as their material, their genetic material, and then they use reverse transcriptase to get that genetic material into DNA so it can incorporate into our genes. So yes, I'm just going to go off track for a second. Please indulge me. There is an amazing thing called an endogenous retrovirus. In our genome, we have entire sequences of retroviruses that are built into our genome. They're lying dormant, they're probably inactive, but they are vestiges of previous retroviral infections that entered the germline of one of our ancestors and now is in all of our species. Holy cow, that's cool. Maxine, is this process similar to HIV virus infection with reverse transcriptase or facilities? Yeah, so I'm not sure about the second part of what you said, but yes, it is similar to HIV and the way that HIV integrates its genome into a helper T cell. That kind of mechanism, if it wasn't in a helper T cell, but it was in a germline cell, that would be a cell in a woman's ovaries and a man's testes, then what would happen is that that retrovirus and the genes that it introduced could become incorporated into a sperm cell or an egg cell, and that person could then possess that retroviral sequence in every single one of their cells. And if that person became one of the common ancestors of all of humanity, then that would become part of the heritage of our species. So really incredible stuff. Vivian, thank you so much for saying hi. It's great to have you here tonight. And in fact, I'm seeing a lot of people here, and I'm really happy to have this level of participation and to have you all here. So we've now talked about conjugation. We've talked about transformation. We've talked about transduction. Are there any more? Let's see on the next slide. There's viral recombination. This is a kind of horizontal gene transfer that happens between viruses with enormous, enormous consequences for humanity. So imagine this, two different viruses, different strains infect the same host. So here's, virus A1, here's virus A2. They're the same kind of virus, but they're different strains. They both infect a pig. Well, they can actually wind up infecting the same cell in a pig. And when they do, during their viral life cycle, they can actually exchange genes with one another. I say it in a way that sounds purposeful, but it's not. This whole viral reproduction cycle, it's quite messy. There's fragments of DNA, there's fragments of protein. And what you wind up having is a new virus that emerges that has a little bit of both of the viruses that infected that pig cell. The genes of the viruses can recombine, and the result is instant emergence of new viral strains. If our immune systems can't recognize the new strain, this can lead to pandemic viral outbreaks. This is called an antigenic shift, where the surface material of the virus becomes something that our immune system can no longer recognize. And when flu pandemics happen, it's because of this. So this is viral recombination. And again, just so that you know about my teaching method and how I think about this, I developed this diagram based on a question that showed up on a former AP Bio exam, because I want people to know about how this is a form of recombination. You'd be able to identify it as such. And, that's going to prepare you for the test. So how are we doing here? That ends the section on biotechnology. Before we go on, any more questions about horizontal gene transfer? We talked about conjugation, transformation, transduction, and viral recombination. Anything that I need to do to delve deeper into any of those topics or clarify them? Okay. All right. Not seeing anything in the chat. Want to emphasize that there's study material about all of this on learn-biology.com. We're actually going to go there in a minute. Hello, gaming. Cool. Does viral recombination and the fact that the new strain is not recognized have anything to do with CRISPR? CRISPR is a different kind of thing. CRISPR is, first of all, it's a natural technology that bacteria use in order to remember previous viral infections and store away information about viral infections into their genome. In the past couple of years, we figured out how to repurpose this natural system called CRISPR so that we can essentially edit genes in a way that's not unlike the way that we edit text on a computer. If you go to learn-biology.com, where would you see this? I think it's on the About page, but I actually did an interview with Jennifer Doudna, who's the recipient of the recent Nobel Prize for CRISPR. That's because she lives in our community, and her son was my student. And she very generously helped out the Berkeley High School parent group. And I got to do an interview with her as part of an event that actually happened during the pandemic. So you can learn more about her. So that's the connection with CRISPR. Hello, gaming. It's not really about recombination in the same way, and it's not really about horizontal gene transfer, though it is cool. It is incredibly cool. On my college site, not really on my high school site, I have a tutorial about CRISPR, and I totally encourage you to learn about it. APUSH warriors, APUSH, APUS history warriors. Is conjugation only in bacteria? There's two prokaryotic domains. There's archaea and bacteria. As far as I know, it's only bacteria that do conjugation. So yeah, if you make that connection, bacteria conjugation, I think that's a pretty safe bet. Good question. Anything else, guys, before we go on? Okay, really appreciating all these questions. These meetings get more and more fun. What's even more fun are the Zoom meetings. And again, $24.95 for a subscription to learn-biology.com. I'm going to put the chat. I'm going to put this right here just for a little. Okay, I can type this. It would be great if, like right now, you called over one of your parents and you said, hey, $24.95, how about the credit card? And I can get a subscription to learn-biology.com. It'd be fantastic. It'd be fantastic for me. I'd be thrilled. And then you could join me on Zoom tomorrow, and you'd have all the resources of learn-biology.com at your disposal. Okay, let's talk about biotechnology, and then we're just going to go onto learn-biology.com. We're going to do some quizzes. It's going to be super, super fun. So here are some of the questions we'll address in this section of our show. What is recombinant DNA? How can it be artificially created? How can you create recombinant bacterial plasmids that express human genes? What is gel electrophoresis, PCR, and sequencing? Some of the stuff in biotechnology you need to know well. Some of them you just need to have a passing familiarity with. We're going to do both in the next 10, 15 minutes. Okay. What is recombinant DNA? How can it be artificially created? So recombinant, you can see the word combine in there. We make recombinant DNA every time we make gametes. And that means that, animals do it all the time. During meiosis, what individuals do is they combine the genes of their parents in the gametes. That's what meiosis is all about. That's when you have crossing over and the creation of recombinant chromosomes. This is different because that happens within a species and it's combining the genes of a parent. What we're talking about is artificially recombined DNA that combines the DNA of 2 or more species. So, oh, and by the way, we're going to be doing this for another half hour. If you have a friend out there in the world who you think would benefit from being on the show, just, do what you do in class all the time. I'm sorry, I'm not a classroom teacher anymore, but like, people were on their phones a lot, when I taught. It was sort of an ongoing battle. And I don't need to go there, but what I want to do right now is encourage you. Text a friend and say, hey, go to Science Music Videos, jump on this live review right now. It'll be super fun. So go ahead, do that. The more people we have, the more questions we have, the more dynamic it's going to be, the more fun it will be. So please feel free to do that. Use your phone. I won't know. I certainly won't take it away from you or do anything that teachers try and do to fight the cell phone war. So what is recombinant DNA? How can it be artificially created? So recombinant DNA is DNA from more than one source. Like, for example, DNA from a bacterium that's spliced together with bacteria with, let's try that sentence again, DNA from a bacterium that's spliced together with DNA from a human and then followed by more bacterial DNA. It's created using enzymes that are called restriction enzymes. That's shown at letter C. What these enzymes are capable of doing is they're capable of riding along a DNA strand and then making a cut in the DNA. You can sort of think of them as molecular scissors. They go along the DNA, they find a specific sequence, and they cut the DNA at that point. They can make a variety of cuts, but the restriction enzymes that are most commonly used make cuts with sticky ends. Why are they sticky ends? Because they have single-stranded nucleotides that are ready to form what kind of bonds? You see, in Zoom, I would just say, what kind of bonds? And people would instantly shout out, hydrogen bonds, Mr. W. But that's what we're talking about. Yeah, we're going to get to that question in just a minute. So those sticky ends are then available for forming new bonds with other pieces of DNA. Now, imagine this. You take the DNA of a second species and you cut it with the same restriction enzymes. It'll have complementary exposed pieces, complementary sticky ends. You put them together in a test tube. It's literally nothing more than that. And this is something that can be done in a classroom and is done. I did it in my classroom. And the DNA will form hydrogen bonds. In other words, this fragment is ready to bond with this fragment. This fragment is ready to bond with this one over here. They'll bond, and that's what's happening over here in this diagram. And then you have to throw in one more enzyme. It's called DNA ligase, and ligase seals sugar phosphate bonds between these fragments. The resulting DNA is, it looks like DNA. It's the double helix, but informationally, it's recombined. It has DNA from 2 species, and you can see that right here. Yera, let me put a link in the chat. Go to learn-biology.com/live/ oh, I always wrote liver, live/reviews. Here we go. So go to that page and you'll see the entire schedule for all the live reviews that we're going to be doing. And Maxime, I covered semi-conservative replication 2 shows ago. I'm happy to do it again, but let me get to the end of this and then we'll see where we are. We'll take a little vote if we want to do semi-conservative replication or if we want to do some quizzing, okay? Yeah, I think if you look on, you can look in a couple of places, Maxime, and I'll show you where you can find all that stuff. So we've just talked about how you make recombinant DNA. Now, what you can do is something really cool. This slide is kind of a zoomed out version of this slide. So what's going on here? You extract a plasmid from a bacterial cell, the same kind of plasmids we were talking about in terms of conjugation. And we're going to use restriction enzymes. They're represented here by actual little scissors to cut that plasmid open with sticky ends. we're going to use the same restriction enzyme to cut out a target human gene. What do I mean? I mean a gene that we want to insert into this plasmid over here. You put them together in a test tube. And because they've been cut with the same restriction enzyme, the human gene will combine with the plasmid, forming hydrogen bonds between the sticky ends. And then, as we showed in this slide over here, you add DNA ligase, you add this enzyme over here, and that will create a plasmid that was that's now a complete and integrated piece of DNA. It's got bacterial genes and it's got human genes. Then what you do is you use the technique of transformation that I kind of described in a previous slide. And what that'll do is now this bacterium has a plasmid with a human gene. That plasmid is DNA. DNA gets transcribed and translated. When this DNA gets transcribed and translated, it becomes messenger RNA, and then it can become protein. Every time this bacterium reproduces, enzymes like DNA polymerase, DNA helicase, all of those enzymes that are involved in what Maxime was asking about, semi-conservative replication, all of those enzymes are going to replicate the plasmid as well as replicating the the main DNA chromosome. And so what we're doing is all of these descendants of this cell will do two things. They'll copy the plasmid, the genetically engineered plasmid, and they'll express the gene. This is how we do things like, this is how we create genetically engineered insulin. Insulin's the hormone that's used to control diabetes. It used to be, the way that it used to be acquired was that animals were slaughtered because our close mammalian cousins, like pigs and sheep and cows, they have a pancreas that makes insulin too. And so we would slaughter the cows and sheep and pigs, and we'd extract their pancreases, and then from the pancreas tissue we'd get insulin, and then we'd purify the insulin, and then that was used for diabetics to treat their diabetes. But now we don't slaughter those animals anymore. Instead, we've incorporated the gene for insulin into bacterial cells, and bacteria just churn out the insulin day and night. They do it in huge vats. It's like making yogurt, but instead of making yogurt, they're making insulin. That leads to some important bioethical issues because insulin should be incredibly cheap, and it's not. And that's about capitalism. It's about the healthcare system in the US and A. It's about a whole bunch of things that are kind of outside the scope of biology, at least AP biology. I definitely have opinions about them, but I'm not going to go into them there because I'm going to try and stay, I'm not going to go into them here because I'm going to try and stay focused. Okay. In order for the preceding process to work, you've got to take out introns. Now, we take out introns all the time during RNA processing, but if you're a genetic engineer and you've extracted the gene for insulin, it might be, it'll have introns incorporated into it. How do you get those out? Well, first of all, let's just review. Introns are non-coding sequences of DNA within eukaryotic genes that have to be spliced out before the gene's RNA can be translated into protein. We do this, I explained it in a previous episode in this series. But to transfer a human gene to a bacterium, you have to get DNA, you have to get intron-free DNA. So here there are two methods. The first is you can actually just take the protein, like the insulin protein, and using somewhat old-fashioned biochemistry techniques that have been around for decades, like I think like back to the 1950s, you can figure out the amino acid sequence of insulin, and then you can just use a genetic code chart and say, oh, we need this DNA, and you can put that together. That's one perfectly viable way of doing it. There's another way that's kind of cooler, which is this. You go into cells that are producing the desired protein, you extract the messenger RNA. It's already intron-free, and then you use reverse transcriptase, which Maxime, you added about before, to create complementary DNA from the mRNA. And then you have intron-free DNA that you can incorporate into the plasmid. Definitely has been an FRQ about this kind of thing, and now you know about it. All right, gel electrophoresis, another technique to know. I'm just checking the time at 7:40. Within the next 3 or 4 minutes, we'll go on to learn-biology.com and we'll do some multiple choice. Practice. Sophia asked a question. Since the DNA came from a human gene, how does the bacteria express the correct gene without doing the RNA processing and stuff? Sophia, this is, I'm not sure, I wasn't paying close enough attention to the comment thread. The bacterium can only express DNA that has already been properly treated so that it's intron free. If this were bacteria, if this were the raw gene for insulin, then the protein that the bacteria would make would have extra amino acids. It would have amino acids that would correspond to the introns. And it's also possible that there could be a frame shift that might happen there. So it wouldn't work. What you have to do is you have to use one of these two methods to get rid of the introns. So Sophia, very, very good question. And Sophie's asking the point of introns. So there are two levels to this question. In bacterial engineering, oh, that's so funny. I did a finger gesture and I got balloons to fly up. That's kind of cool. There are two levels to the question. In bacterial engineering, the introns are just a pain in the you-know-what, and they have to be removed, and they can be removed by this method. Why do we have introns in our genes? It's a complicated question. One is that, I'm thinking about what are the answers to why there are introns. One is that the idea is that the introns are kind of selfish bits of DNA that are just trying to do what any DNA does, which is make more copies of itself. So it's not about our purpose, it's about their purpose. But there is another thing. Because we have genes with introns, and I discussed this 1 or 2 nights ago, when proteins are assembled, you can have alternative splicing of exons. So like, for example, here we have 4 exons. You can make a functional protein that might just have exon 1, 3, and 4. Or you can have one that is exons 1, 2, and 4, and another one that's 1, 2, and 3. And those might all be related proteins that do different things. So the presence of introns allows for something called alternative splicing, and that increases phenotypic variation among proteins. So that's the basic idea. Maxime, I think you were trying to respond to that. And that's a pretty good response. But really the key term, I'm going to type this in the chat, is alternative splicing. So I have a tutorial about that, or that includes that on learn-biology.com. I'll show it to you in a minute. We're just going to jam through the rest of these. Gel electrophoresis is how you separate pieces of DNA by size by running fragments through an electrical current. The basic idea is that large pieces of DNA will move more slowly than small pieces of DNA. You wind up with something like this. No, I didn't label this. Here are the large pieces, here are the small pieces. If these were 3 different individuals, then you could tell, you could look at their different pattern. So this is widely used in forensics. It's used in all kinds of analysis of different species. It's a go-to technique. So gel electrophoresis separates DNA fragments by size by running them through an electrical current used for DNA fingerprinting. I'm going to skip over this problem right now. I just want to mention these other 2 techniques. PCR is used to amplify DNA. It's a cell-free way of amplifying DNA in a test tube. It stands for the polymerase chain reaction. And basically, through a combination of heat-resistant DNA polymerase primers, excuse me, and free nucleotides. You can take DNA, you can put it in a machine, you can amplify it a billionfold in 30 minutes. So this is widely used, again, in any science where you have to analyze DNA because you need a significant amount of it. And then finally, the last technique to know about is sequencing. And sequencing is using actually a modified form of, it combines PCR and electrophoresis so that you can actually determine what the base by base sequence is of various of genes, sequences of DNA, or the entire human genome. You guys are too young. You were born after this, but while I was alive, because I've been alive for a long time, the human genome was deciphered. So this happened in the 1990s. It sort of culminated in the late 1990s. The entire sequence of DNA for a human being was deciphered. So this is the result of sequencing. And you can do cool things, like you can take a sequence and you can say, oh, human sequence is like this. What's a gorilla sequence like for that same? Oh, and it looks like, oh, the gorilla and the human have the same sequence. What does that tell you? That we have a recent common ancestor. So it's widely used in evolutionary analysis. It was used to figure out the SARS-CoV-2 virus. It was used to create the vaccine. It's a remarkable technology. The whole field of informatics, bioinformatics, is based on sequencing. Okay, let's do some, I'm hoping you're going to hear the sound for this. Oh no. AP Bio Review. Multiple choice questions. I hope you heard that. I really hope you heard that. That was wonderful. Okay, so you don't have to type this in because I'm going to type this in. I'm just going to go to, I'm going to show you how I do this. I'm going to go to learn-biology.com. Okay, so this is my website. Just want to show you something. If you want to learn more about what this site can do for you, click the student link over here. You'll go to our student page, and that's where you can get started. For $24.95, you can buy a subscription. But right now, what we're going to do is we're going to go like this, and we're going to hit this link over here, AP Bio Review. You can do this once you have well, you can do it right now. And let's go to our multiple choice review. And let's say that you've gone through our checklist and let's say that you have decided, oh, I need to practice a little bit about horizontal gene transfer, mutation, stuff like that. So here's what I'm doing. I'm expanding gene expression and regulation, unit 6, and mutation, this should say mutation and horizontal gene transfer and biotechnology. Those are the two topics that we just covered. That's what we want to focus on tonight. So we've got a total of 18 questions in there, and let's begin practice. So we're pulling down the questions, and we're getting a question about operons. And I'm going to expand this a little bit so we can see it. We didn't do this tonight, but we did this 2 nights ago, so why not try it? A long question, but let's just do it, and we'll see how well you guys need to understand operons. And again, here's the deal. If you find that you don't understand operons very well, then what's the thing to do? Go to learn-biology.com, do our tutorial about operons. The diagram below shows the lac operon in the presence and absence of lactose. The genes LAC-Z, LAC-Y, and LAC-A allow the bacterium to use lactose as an energy source. Okay, clear enough. A researcher adds a gene for ampicillin resistance, AMP-R, between the LAC-Z and the LAC-Y genes. So here's the genetically modified sequence over here. Then the researcher plates the bacteria on the following media. All media contain fatty acids as a secondary energy source. Bacteria, when given the choice, metabolize sugars before fatty acids. Pars. I'm working on that right now. I can show you some good alternatives, but I'm a little bit behind where I wanted to be, and I'm struggling with that. So then the researcher plates the bacteria on the following media. All media contain fatty acids as a secondary energy source. Bacteria, when given the choice, metabolize sugars before fatty acids. So plate 1 has no ampicillin or lactose. Plate 2 has ampicillin and no lactose. Plate 3 has lactose and no ampicillin. And plate 4 has ampicillin and lactose. So is everybody getting what's going on here? Researcher has modified the gene for the lac operon so that it has this AMP-R resistance. That's ampicillin resistance. By the way, if you're thinking this is a tough question, this is modeled after an AP question. So the question is, which plates would there be growth on? What do you think? Would it be plate 1, where there's no ampicillin or lactose? Would it be plate 2, where there's ampicillin and no lactose? Would there be plate 3, where there's lactose and no ampicillin? And would there be plate 4, where there's ampicillin and lactose? So it's confusing, it's difficult, it's a multiple choice question, and you have to make a choice. So I'm going to set this up so that you can see as much of the question as possible. And now you got the whole thing. You've got the whole thing on the screen. I guess I got to go like this. Yeah, there we are. So, okay, Maxime is saying 1, 3, 4. Lily is saying 2. Sophie is saying 1, 3, 4. Sophie, very nice to hear you chime in, by the way. Well, let's go with nice job, guys. The bacteria will be able to grow on the 2 plates where there's no ampicillin, plates 1 and 3, right? In addition, the presence of lactose will enable the bacteria to express the gene for ampicillin resistance, so they'll be able to grow on plate 4 as well. If you did a transformation lab, usually this is the kind of array of plates. And again, I've got a tutorial about this on learn-biology.com. I know that I say that a lot. Let's do another question. Very good. Similar. The image below represents the piglo plasmid used both in research and in education. Okay. As indicated, the plasmid contains a restriction site. That's a site where you'd put restriction enzymes. Now I'm regretting skipping this question that I talked about before. Contains a restriction site and 3 genes. 1 gene is for AMP-R, that's ampicillin resistance. We just talked about it. GFP encodes the green fluorescent protein, GFP, which glows green. It fluoresces under ultraviolet light. And ARIS-C encodes a protein required to promote the expression of GFP when arabinose, a monosaccharide, is present. The results of a bacterial transformation experiment using the piglo plasmid are shown in the table below. All right, so these are bacteria that were not transformed with the plasmid. These are bacteria, they're growing just on plain nutrient agar. These are untransformed bacteria that are growing on nutrient agar plus ampicillin. These are transformed bacteria that are growing on agar, ampicillin, and arabinose. And these are transformed bacteria that are growing on agar and ampicillin. What do I mean by transformed? They took up the plasmid. Okay, these did not take up the plasmid. So which of the following statements about the purpose of plates W or X is correct? All right, I'll just read them. Plate W showed the successful destruction of the plasmid by bacterial restriction enzymes. 2, plate W showed that there was extremely high transformation efficiency. 3, plate X showed how nutrient agar can enhance the growth of viable bacteria. 4, plate X showed that ampicillin was effective in inhibiting the growth of bacteria that had not taken up the plasmid. Which of those 4 is the answer? This is an experimental design question, so important to know. 1, 2, 3, or 4. Got to make a choice. Okay, see one, see another. Autumn, haven't heard from you yet. Welcome to the game. Welcome to the game. Sophia, fantastic. Maxime, okay, nice. All right, everybody saying 4. The answer is indeed 4, but I just want to show you something. Let's say that you got confused and you didn't know, and you chose this over here. Well, you'd get feedback. It would say, no, there's no growth on plate X. And that's due to the presence of the antibiotic. Next time you see this question, think about the idea of experimental controls and make a different choice. So what I do, and the reason why I'm saying I is because I wrote this for my students years ago, not really many years ago, is because I give hints, and then you think about it, and then you see this question again, and then you learn it much, much more deeply. 4 indeed was the answer. Good job to all of you. Let's do another question. 2 cloning vectors used in genetic engineering are bacterial, viruses, and plasmids. Which of the following statements about viruses and plasmids is false? So one of these statements is false. The first question, plasmids use the translation machinery of the cell. 2, viruses have a protein capsid. 3, viruses can replicate in the absence of a cellular host. 4, plasmids carry genes. 5, plasmids have restriction sites. So which of those statements is false? 1, 2, 3, 4, or 5? Unbelievable. Okay, and indeed that's true. Viruses are obligate intracellular parasites. I'm going to put that term in the comment thread. Obligate intracellular parasites. They rely on cells in order to carry out their life cycle. So viruses can't replicate in the absence of a cellular host. Folks, I'm going to jump back to our slideshow because I want to do this or this. I didn't want to do that. Okay, couple of things to do. Do them now if you haven't already. Download the checklist. It's at apbiosuccess.com/checklist. It's a great tool for you to review. Second thing I want you to do is this. I want you to think about 3 reasons to subscribe to learn-biology.com. And what I'd really like is for you to do that subscription right now. Just call over your parents. Hey, mom, dad, help me out. I need the credit card. And basically, here's the reason. First of all, you get free Zoom tutoring. If you liked this experience tonight, what's going to happen tomorrow night is 100 times better. So you get me. You get me live for $24.95, and that's all I'm ever going to charge you for it. And again, it's just $24.95 because you're going to cancel your subscription at the end of the month. So $24.95. I'd like to see that happen now. Call over your parents, get them to do that. Jitsu, I am so thankful to you for coming. And what I'd like to see is, I'd like to see you, please get a subscription to learn-biology.com so you can get the Zooms. Another thing is you get incredible support 24/7 on learn-biology.com. Think about what would happen to your understanding if you did the kind of quiz that we just did together for 45 minutes every single day between now and the AP Bio exam. You'd get so much feedback about what you know and what you don't know. Your understanding would improving. You would do so much better. So again, call your parents over. Go to learn-biology.com/AP Biology. Just hit the now button and you can join, you can get a subscription, and it's going to totally change your experience between now and May 16th. The third reason to subscribe to learn-biology.com is that you get the Biomania AP Bio App, which I would show you, like I'd hold it in my hand, but I'm using my hand as my webcam right now, so I can't, but I have a bunch of screen grabs. Basically, you get a lot of the interactivity that's on learn-biology.com, but you get it on your phone. You can study anywhere. You can study on the bus. You can study when you're doing anything. You can be comfortably lying in bed. You can study, and again, it'll change your experience. So I'd love to see, there have been up to 14 people who have been with me tonight. I'd love to see, I know that some of you are already subscribers, but I'd love to see others who aren't subscribers yet to go to learn-biology.com and subscribe. It's going to really change your experience of studying. Everything that I know about biology is there on learn-biology.com and the Biomania AP Bio App. So I want to see you at the next review session. I want you to promise to invite a friend. The next Zoom review is tomorrow night. Spread the word. Spread the word, everybody, and subscribe to learn-biology.com. Do it now. Go to learn-biology.com/apbiology, and I really want to see, and I've got a couple of people here who are saying see you tomorrow. That's because they paid $24.95. You can do the same. I'm really looking forward to getting more subscribers and helping more people. So I hope that you're calling over your parents and you're saying, Mom, Dad, $24.95. It's not nothing, but it's not a lot either. And it's going to help me deeply. It's going to help me substantially as I prepare for the AP Bio exam and help me achieve the success that I crave. So folks, it's been fabulous. I'll see you again in a couple of days. I'll see some of you on Zoom tomorrow. I want to see every single one of you on learn-biology.com. A-Push Warriors, I want to see you. Jitsu, Sophia, Maxime, go to learn-biology.com/AP-biology. I'll just leave it up for another second. learn-biology.com/AP-biology. Subscribe right now. It's going to be the best decision you've made in terms of getting yourself ready for the AP Bio exam. Thank you all so much for joining me tonight. I'll see you at the next session. Everybody, let's see if I do this. Do I get like balloons or something like that? Everybody, be well. Have a great night. Thank you so much for joining me.